Now that we have explored the anatomy of the digestive system, we will review the physiology of enzyme digestion and how absorption of the digested nutrients takes place. So recall that digestion refers to the breakdown of complex food molecules into their smallest chemical building blocks, that are called monomers. These monomers include monosaccharides, come from the breakdown of carbohydrates, amino acids that come from the breakdown of proteins, and fatty acids that result from the breakdown of lipids and fats. The mechanism of chemical digestion occurs by enzymes. The full name of this process is enzymatic hydrolysis. This means that the enzymes break chemical bonds and add water to the sites where molecular bonds are being broken in order to stabilize the monomers that are created. We see that enzymes are typically secreted into the lumen of the alimentary canal, but a few in the small intestine are found on the brush border of the microvilli. Also recall that the primary site of enzymatic digestion is going to be the small intestine. Absorption is the movement of the digested materials from the lumen of the gastrointestinal tract into the body fluids. Just like the main site of digestion was the small intestine, we also see that the main site for absorption is going to be the small intestine, as the small intestine is equipped with the large surface area and the blood vessels that will promote very efficient absorption of those nutrients. So recall that the epithelia of the small intestine are joined by tight junctions. This means that materials cannot move between cells, and instead it has to move through them. The blood from the intestinal capillaries that picks up these nutrients will send those nutrients to the liver through the hepatic portal vein. And we see that most nutrients, so that is particularly monosaccharides as well as amino acids, are going to be absorbed by active transport, which is going to require some energy, some cellular energy, which in this case is ATP. Lipids can diffuse directly across the plasma membrane using simple diffusion because lipids can go directly through the lipid part of the membrane. Starting with carbohydrate digestion, reviewing it, we'll see that carbohydrate digestion begins in the mouth with an enzyme called salivary amylase. We see that it is finalized in the small intestine by pancreatic amylase from the pancreas. And some carbohydrate digesting enzymes are also found on the brush border of the small intestine as well. The names of the specific brush border enzymes are not particularly important here. So we see that the small intestine can only absorb monosaccharides, that is galactose, glucose, and fructose. And so any undigested carbohydrates, like plant fibers, will not be absorbed, but will help keep food moving through the alimentary canal. Brush border enzymes will digest disaccharides, like lactose, maltose, and sucrose, into their component monosaccharides. Lactose is digested into galactose and glucose. Maltose is digested into two glucose molecules, and sucrose is digested into fructose and glucose. From there, they can be absorbed across the enterocytes and into the bloodstream of the capillaries that are located in the lamina propria of the small intestine. So keep in mind that the intestinal enzymes tend to work best in alkaline conditions, typically around a pH of 8. Pancreatic juice has an abundant amount of bicarbonate in it, so that the pancreatic juice has the ability to raise the pH of the chyme that is entering the small intestine from the stomach. Two terms to be aware of here while we look at the absorption of nutrients, specifically at the enterocytes where absorption of nutrients is going to take place across, is that we have the apical membrane of the enterocyte and the basolateral membrane of the enterocyte. The apical membrane is part of the plasma membrane on the enterocyte that faces the lumens. That's the part of the plasma membrane where nutrients are going to be absorbed from the lumen of the intestine into the enterocyte. Well, the basolateral membrane faces the capillaries that are located within the lamina propria. So once starches and disaccharides are broken down into their component monosaccharides, the monosaccharides are then actively transported across the apical membrane along with sodium. So once inside of the enterocyte, the monosaccharides are transported across the basolateral membrane of the enterocyte by a monosaccharide carrier, which is a form of facilitated diffusion. So the transport that we see on the apical membrane is a form of secondary active transport, and the movement across the basolateral membrane is going to be facilitated diffusion. So once the monosaccharides move through the monosaccharide carrier on the basolateral membrane, they can then diffuse into the bloodstream and be carried away. So one thing that we see as we age, the enzyme that digests lactose, which is called lactase, tends to decrease in production. We see that excess lactose will remain in the small intestine and create an osmotic pull that causes water to remain in the small intestine as well as in the large intestine, which causes diarrhea as well as gas. We say that gas comes from the bacteria metabolism of the undigested lactose. So that is, bacteria in the large intestine are breaking down the lactose that was not absorbed by the small intestine. And so in the process, they're creating gases and other irritants. So we see that drugs like lactate contain lactose enzymes, which help us to digest lactoses, which come from dairy products. Reviewing protein digestion, we see that, or recall that protein digestion begins in the stomach with an enzyme called pepsin. See that pepsin functions optimally at a low pH, that is a very acidic pH of about 2. So that's why acids like hydrochloric acid are present within the stomach. Digestion of proteins is then finalized by the small intestine by enzymes like trypsin that comes from the pancreas, as well as a number of brush border enzymes on the surface of the enterocytes that will break down those small protein pieces into amino acids. So again, here the specific names of the brush border enzymes are not important, but do recall that the enzyme that digests proteins that's secreted by the pancreas is called trypsin. So just like we saw with monosaccharides, amino acids are actively transported through the apical membrane along with sodium. Once inside the enterocyte, the amino acids are transported across the basolateral membrane of the enterocyte by an amino acid carrier, which again is a form of facilitated diffusion. So the amino acid will then enter the bloodstream and be carried away. Finally, digestion of lipids really does not begin until the lipids reach the small intestine. We did see that the salivary glands produce lingual lipase and the stomach produces gastric lipase. But these enzymes do not function much in the digestion of lipids because bile is not secreted into any of these regions. So recall that bile is necessary to help break up the lipids like a detergent so that the lipases then have a greater effect on digesting them. We see that the bulk of lipid digestion occurs in the small intestine due to the fact that both bile and pancreatic lipase are present. We see that the bile once again is emulsifying lipids, and emulsification is simply mechanically breaking down the lipids into smaller droplets, which gives the lipid more surface area so that enzymes like pancreatic lipase can more easily and more efficiently digest them. So lipases will digest lipids into monoglycerides and fatty acids. Because lipids can diffuse directly across the plasma membrane, we don't need a carrier protein in order to get them into the enterocytes. So we also don't need ATP energy to do it as well. So those lipids will simply diffuse through the enterocytes, and once they are inside of the deeper tissues of the villi in the small intestine, they will enter the specialized lymphatic capillaries that are called lacteals. And those lacteals will then carry those lipids to the liver for processing. So recall that the secretions from the salivary glands, the stomach, the pancreas, and the small intestine require a large abundance of water. So as a result, approximately 9,000 milliliters of water enters the small intestine each day. Some of it is the water that we've consumed, but most of the water is coming from those digestive system secretions. So those include the salivary gland secretions, gastric juice, pancreatic juice, intestinal juice from the small intestine, as well as bile. We'll see that 95% of this 9,000 milliliters is absorbed by osmosis in the small intestine. Any remaining is absorbed by the large intestine, also again by osmosis. we'll see that approximately 100 milliliters will remain in the food residue in order to soften the stools. So to summarize, most nutrients, by that I mean carbohydrates as well as amino acids, are absorbed by active transport. So that is, those require ATP energy. Lipids, because they can diffuse directly through the lipid part of the plasma membrane, have the ability to diffuse passively, so that is by simple diffusion, across the plasma membranes of the enterocytes and get into the lacteals. We see that the plasma membrane of the enterocytes in the small intestine also has protein channels that allow for the diffusion of water into the bloodstream by osmosis.